of Unit 5. And first of all, Unit 5 is a chapter which is not known to many of our students, okay? And because Unit 5 is not exactly integral part of most of the courses like microbiology, biotechnology, it's not included in that college course and curriculum. That's one problem. So what we will do here is that Unit 5 is nothing but an extensive knowledge of cell biology and molecular biology. Okay, because you will see that in different aspects of development biology, we will see the applications of cell biology extending the idea of cell biology and molecular biology. So if you are good enough and if you have already prepared molecular biology and cell biology well, then you can catch the concepts of development biology very well. So it's very important that you prepare cell biology and molecular biology prior to the preparation of Unit 5. That's why it's provided in Unit 5. Okay, so let's begin with today's class. In today's class, we will be talking about the basics of development biology. Means we're not going to talk independently about any single organism and its development, but we will take different examples while explaining the different part of the development feature and embryogenesis. That includes the sequential events starting from the fertilization, then the cleavage, and afterwards gastrulation, and morphogenesis, metamorphosis, and all those things. So we will talk about the different features that are present in a cell while it's developing from an embryo, developing from that single cell that is a zygote. So we'll talk about all of that. Okay, so while explaining all these concepts, I will also use the help of different animations that I have listed, and today I'm going to show you 8 to 9 separate animations explaining each of the different phenomena correctly, so that you can understand the mechanism fast and in better engaging way. Otherwise it will be difficult to understand. Okay, so let's begin. So the first thing is the overview and the components of development. So what I can tell in a very simple terms, what is development? The answer to that can be explained with this: a series of progressive changes that generates the structures of the organism throughout its life cycle. That is known as the organism's development. Okay, now the series of change occurred over a period of time. Okay, so that series of change starts from a single cell and then it's modified. That single cell is modified into multiple cell. How? That single cell continues to divide. It continues to divide and producing many cells. And when it produces multiple cells, let's say a single cell that was known as the zygote divided, and now it is 8-cell structure. So that 8-cell structure that they prepared, now from that 8-cell structure it continues to divide and produce another structure, as you can see in the bottom of this picture. So zygote is the one that we all begin with. Okay, so if you take it as an example, like echinoderms, like a starfish, they also start from here. You take sea urchin, they start from there. A frog started from there. A chicken started from there. A human also starting from that same place. Now from there continues cell division, producing a mass of cell which is also known as modular stage. Okay, which is a rigid mass of cell, remember. Now those mass of cells, all those cells start to separate and present in the periphery, and the center part becomes a hollow region which is filled with fluid. This is known as a fluid filled cavity, and this state is known as blastula state. Okay, so once the blastula state is formed, then it is being modified. Okay, how exactly? Now those cells that surrounds that fluid filled cavity start to migrate towards a specific direction, and this migration will lead to the formation of cavity which is known as the gut cavity. Remember, because we all have the gut cavity in there, because in few organisms all those organs they are floating in the gut, and some other organisms we have specific structures and organization to hold them. Here we call them the coelom. Remember, because few organisms have a complete coelom, few organisms have only pseudocoelom, and few organisms are acoelomate because they don't have any coelom. Now, another thing is that this gastrulation is slowly modified to produce larva. Larva stage is not present in all organisms. Here we are looking at the organism starfish. In that case, they have the larva state. Then finally producing. So while the cells are migrating, that state is known as gastrulation. So this is blastulation. This is morula. Okay, and this is the zygote. And before the zygote even, we have two cells that forms the zygote. This is known as the fertilization. Fertilization is a process where the sperm donates its nucleus inside the egg, and that completes the formation of a zygote. So we'll see the sequential events that includes the fertilization. Then we'll talk about this morula, which converts the zygote into the 8-cell step, and this conversion is nothing but a rapid cell division. And this rapid cell division we are talking about, this rapid cell division is also known as the cleavage. Okay, and then morula converted to the blastula state, blastula. So this is second, morula, then blastula third, gastrulation fourth, and finally the organogenesis, that means structures generated for all the parts of the body, and finally we make the adult organism. So these are the sequential events as the definition said. Now the sequential events, we'll talk about independent events. We'll discuss about each of these events separately in today's class. But these are not just independent events that are occurring, but also these events have meaning. Means, you know, while these events are occurring, they're cross-talking with themselves. A cell from one part of the body is cross-talking with the neighboring cell. It's allowing the neighboring cell to produce a specific structure. So there are few features of the development or developing tissue. That includes a rapid growth, that includes differentiation, and also morphogenesis. Now growth is a change in size due to the increase in the cell number or the cell size. Differentiation, where it is known as a cellular specialization. That means while we are talking about cells growing, we know that all of us started the journey from a single cell, a zygote. Now the zygote, when start dividing, producing many different types of cell. Now all those different types of cells that a zygote is preparing, now those cells are non-specific. They are not specialized to do any of these tasks. So they are just randomly placed. Now those cells will start to do a specific function in the body, in the adult organism. For example, in our body we know that few layer of tissue is going to produce our nerve system only. They are not going to produce the liver. They are going to produce the neurons. On the other hand, there are some tissues that are going to produce the muscle. They are not going to produce the eye, lens. Okay, so they are determined to do a specific fate. That is the fate. So fate differentiation is not done at the very beginning. It's done a later stage of the development, but it is very, very important. And then morphogenesis, that is establishing a solid body plan. Because you know, you cannot build anything if you don't have a blueprint of that stuff. You need to have a blueprint, you need to have a sketch. Now using that sketch you will develop something. The same thing works here, that to produce an organism you need to have a body plan. Now according to the body plan, you'll start adding different cells, and those cells will cross-talk between themselves for their function, and they actually achieve a particular function. Okay, now among these three features, growth is very easy to understand. We know about the cell cycle, so cell division. We need to understand more about the differentiation and morphogenesis in this chapter very much, because we have already understood the cell division process earlier. So this differentiation is really, really important. It's one of the unique feature of embryogenesis, which is not present in any other part of the cell division or cellular behavior. It's only when multiple cells are working together that they're finally determined to produce a certain type of cell. So this determination means commitment of a cell to a particular fate occurs by activation of specific genes. Obviously, if we need to form something, if a cell need to modify itself, need to produce a particular function, definitely it needs to interact, it needs to be regulated by the gene. So obviously everything is present in the gene to be controlled. but the DNA material that is present from before, it's not the distribution pattern of the DNA is not the same for every single organism. Okay, so for different organisms, the distribution of their genetic material is different in the zygote there. Okay, so that's why, you know, few organisms have like for example, if you look at this experiment where we're looking at the 8-cell stage, and what we can do is that we can separate 4 cells from each other with a glass needle, either horizontally or vertically. So if you separate the cells horizontally, what we can see here is that we separate them 4 cells apart in each side. Now each of these 4 cells, one part of the cell remain embryonic, the other part of the cell produce abnormal larva. So neither of them produce an adult larva at the end. So both of them fail to do so. While if you separate that same 8-cell stage vertically, then it results in a normal larva, although the larva is smaller in size, but it is normal. Now what this experiment can tell us, this experiment is suggesting that there is some material needs to be present and needs to contact both the hemisphere. If we think of these two as a separate hemisphere, the top one and the bottom one, then both the hemisphere need to contact with one another for the larva to develop. So while in this case, if we separate horizontally, there's no way that both the hemisphere is contacting with themselves, so that results in abnormal larva. While if you separate them vertically, it separates it properly, so it gets both the cells from the hemisphere, and that results in a normal larva, although small in size because the cytosol is reduced by half. So this determination is affected by the cytoplasmic segregation, the example of which we just saw in this experiment. And induction is another unique feature that is required while an embryogenesis is occurring. Induction is a process where one cell influences, okay, another neighboring cell to behave and form the similar type of tissue. Okay, that is another unique feature which we will discuss in the next slide. Now this determination, we know that once a cell is determined to produce a certain organ, that leads to the formation of differentiation between the types of the tissue. Okay, there are different tissue types are determined they're differentiated because they are determined to have a particular structure. Now what was that induction? Induction is a process in which a substance or tissue influences the fate of a group of adjacent cells. Normally, for an example, while embryogenesis is occurring, one cell which is producing our brain, okay, a layer of tissue is producing our brain, they will influence the neighboring cell to produce some part of organ or any other cell system which is related to the brain. For example, here you can see during the development of the frog head, what we can see is that this is developing forebrain, and the developing forebrain is surrounded by this surface tissue outside. So while this developing forebrain is growing, this forebrain tissue is actually pushing this external membrane, external layer, which is supposed to form a lens placode and ultimately forms the eye lens. So forebrain structure influences the neighboring cell to modify its structure to produce a lens of the eye. So what we can see that this developing forebrain, it arranges itself in a way so that the lens, the surface tissue start modifying itself, okay, and it start modifying itself to produce lens placode tissue. And what kind of modification? If you look at here in this picture, the cell shape is changed. Earlier the cell shape is something like, you know, cuboidal, like something like this. Now the cell shape is elongated and straight, something like this. It's being altered, and then not only the shape has been altered, but also as this lens placode start to create an infold to form what is known as an optic cup, because it looks a cup shaped structure. This optic cup allows the arrangement of the layer of this lens placode tissue, and then finally both this side will come close and it will join itself to form what is known as an eye lens. And outside of this eye lens they form another layer which becomes the cornea of the frog eye. So this is one simple example of how this frog head and developing forebrain tissue is influencing the surface tissue to form a lens. Okay, so the surface tissue at this point of time, while it started the journey, don't know what it is, don't know what it needs to form. It's not determined to do a particular structure. But when this developing forebrain start releasing chemical components, signaling molecules that will allow and tell this surface tissue to modify itself and produce the lens. Okay, so whenever we are talking about this idea of induction, induction always take place in terms of cell signaling. Another very common example of induction is the vulvar development in C. elegans, from where NET asks a lot of questions. So we'll talk about that also. In the vulvar development also you will see that one cell is at its core. So one cell which is known as the anchor cell. The anchor cell started the process of releasing a chemical component, and this chemical signaling molecule binding to the nearest cell, and the nearest cell also starts to signal and tell the neighboring cell to form the vulva or egg laying structure in C. elegans, which is also an example of induction. We'll see that mechanism in details later. So now remember, these are the process, all the cells that are present, and the cells, while from the single cell the cell is dividing. So while the cell is dividing, they need to share the information, because at the very beginning, at the very early point, the cell carries both. It carries the cytosol, it also carries the genetic material, but the cytosol component needs to be shared. So the more the cell divides, the chance of sharing this material is more. So it's sharing that material with the neighboring cell, and while transferring it to the neighboring cell, they're cross-talking with themselves, and finally they involve in that interaction that causes the determination. And while they establish the determination to produce a certain organ, then they start working together, because that's ultimately what it's all about, a living organism. Because a living organism does not necessarily mean few cells living and sticking to one another. It's all the cells sticking to one another, forming a cell organ system. Organ system means obviously those organs are correlating with one another, they're working together, so one organ is linked with the function of the other organ, and things like that. Okay, which is also the same thing at the cellular level. One cell is involved with the neighboring cell for its process, for its function, and it's required because all of the cells work together to produce an adult organism. So now we will start talking about the sequential steps of the development process. And remember one thing, the question you can expect from the part that is fertilization. You'll get a question from fertilization. Second thing, you'll get question from the adhesion or the cleavage process, also one question from there. You'll get a question from gastrulation, one or two questions from gastrulation actually, and modularity in Drosophila, one question, one or two. You can also get a question from the determination process or induction process at the end, one question from there. So almost 5 questions you will get from the animal part of this development biology. So again, while you're preparing, these are the sub-unit distribution of the questions that you will expect from unit 5. So let's start with fertilization event. In fertilization, they ask either group B or group C question, depending upon the type of question that they want to ask. Now, first of all, as I said earlier, that we're not going to stick to any particular model organism and explain that process of development in that organism. We'll talk about the development overall, and while talking about the development, we will discuss that few model organisms work as best model to explain fertilization. Few model organisms are used better way to explain, let's say, other things like the gastrulation. So like fertilization is very well understood in sea urchins. We'll take sea urchin as a model organism here, and induction is very well understood in C. elegans. Analysis of genes and how they interact is also very well understood with C. elegans. We'll take that. Modularity is very well understood. Organogenesis is very well understood with Drosophila. So we'll take Drosophila as a model organism there. And the gastrulation is very well understood in all chicken, frog, and sea urchins. We'll take all these three and we look for all the differences. So they can ask you question from any of these model organisms specifically, although there are minor differences with the process. So the process of fertilization is always need to be explained with this sea urchin fertilization, and in this process it is a sequential event itself, because here you can see that the process begins with the binding of sperm to the out or extracellular matrix. The extracellular matrix are different names for different organisms. It has zona pellucida name in this case. So binding of this sperm to the zona pellucida, and you know, if you look at the structure of the sperm, it contains mainly three components here. One is the acrosome, that is if I draw the structure, it will look something like this. This is the collar and the rest of the tail of the sperm. Okay, so this is acrosome. This is where the nucleus is present. This is the collar where the mitochondria is present to provide energy. So this acrosome is very, very important, because acrosome is the place where it carries a lot of vesicles filled with hydrolytic enzymes. Because you know, the egg has a layer outside. You know, for example, in sea urchin, they even have that the jelly coat outside. They need to destroy the jelly coat. Then finally they encounter with the cell membrane of the egg, and they need to destroy that as well, rearrange that as well, and then finally inserting the nucleus inside. So what we have here, the acrosomal reaction is very, very important. This structure is known as acrosomal process because it carries a lot of, you know, the modified version of the end vesicles which are formed from the Golgi apparatus filled with hydrolytic enzymes. So once they are in contact with this zona pellucida, they release all those acrosomal vesicles. Vesicles will fuse with the membrane, and all the hydrolytic enzymes are being released. Those hydrolytic enzymes start to degrade the zona pellucida. So as they are degrading the zona pellucida, they start penetrating the zona pellucida, and finally the sperm with the head, they are in close contact with the egg cell membrane. Now once they are contact in the egg cell membrane, then they rearrange their membrane structures like a membrane fusion, and we know membrane fusion is kind of easy to achieve. It's simple rearrangement of the lipids. So once they rearrange the membrane between the egg and the sperm, then they release the nucleus inside. So once they release the nucleus inside, that's the end of fertilization. Active fertilization is done, but a secondary response, a second part of the fertilization still remains. The fertilization is a process where it states the idea of reproduction between two organisms, right? Now we know that in the last lecture we discussed about the speciation, and we know that organisms from different species, they never reproduce between themselves. It's not possible. Now why? Because I told you that there are pre-zygotic and post-zygotic barriers, and one of such pre-zygotic barrier is this receptor that is present on the surface, because the sperm that is released from one species will not fertilize the egg from other species. It's not possible, because for the binding of the sperm with the egg, they need a proper specific sperm receptor on the surface of the egg, and that is known as a binding receptor. So binding receptor present on the surface of the egg, and binding molecules are present on the surface of the sperm. So this binding molecule will interact with the binding receptor, and this specific interaction causes the one signal. That signal is really, really important. That signals that egg cell that now proper sperm from the same species have initiated the attachment. They will do the process of nucleus delivery. So afterwards they should initiate a process known as polyspermy prevention. Because polyspermy prevention means one egg must be fertilized by one sperm, not more than one sperm. It's not allowed. So once one sperm properly delivers its nucleus, that's done. So that particular organism will not allow any other sperm to interact. Okay, so what will happen now, right after the binding, the specific binding, and the sperm delivers the nucleus inside, the egg, then they start to trigger a lot of reaction known as acrosomal reaction. What is this acrosomal reaction in really state phase? Because acrosomal reaction we have talked earlier as well, the release of all those hydrolytic enzymes that will destroy a lot of proteins in the extracellular matrix. Here are the extracellular matrix proteins that we can see in mammals. This is an example in mammals. Earlier example we saw here is in sea urchin. This is the example in mammals, where the mammals have the mammalian egg, they have the layer outside known as the vitelline layer, right? So what we can see here is the altered structure of the zona pellucida that we can see. So whenever the acrosomal reaction take place, that will destroy many extracellular matrix protein, and few of them like ZP2, zona pellucida 2, zona pellucida 3. So ZP3, ZP2, these are the structures that were present. They form these ECM structures outside the egg, or ZP1 as well. Now, due to the release of this acrosomal content, acrosomal process, ZP3 only survives, ZP2 is destroyed, ZP1 is also modified. So as a result of which, it will this change, this modification will not allow. It causes few things. It causes few things. For example, the contact and fusion of egg and sperm to the plasma membrane is prevented afterwards. Why? Because it causes few modifications. It causes a hardening of the cell layer outside the egg. Okay, right after the process of acrosomal reaction. Okay, so once the successful delivery of the nucleus is done, there is alteration of the structure of the ECM that causes the change in the outer membrane of the egg, that makes the egg less permeable by any other sperm, so no other sperm can do the same process later. Okay, now the idea of polyspermy. So this process of acrosomal reaction that we discussed earlier, this is one step towards this polyspermy prevention, right? So polyspermy prevention is done by two different stages. One is a fast step or transient, fast but transient step. The other one is slow but permanent polyspermy prevention. The fast prevent the sperm fusion. The slow or the permanent one remove any other bound sperm. Because remember one thing, lots and lots of sperm started their journey towards the egg to fertilize the egg. So at this point, when we are talking about how it's working and all, we know that not only one sperm is enough and capable of doing the process of fusion, but maybe 2-3 different sperms can do that. They can at least attach with the egg surface and can start to initiate the nucleus delivery, but it needs to be prevented. Now let's say once one sperm started the journey, let's say at one point, this is the egg, one sperm, 2 sperm, 3 sperms are attached, and neither of them delivered the nucleus yet, so they can allow to attach and remain attached to the egg. But now if one successfully delivered the nucleus, now let's say this is the egg nucleus, this is the sperm nucleus. So if one successfully delivered the nucleus, then rest of the sperm should be destroyed and actually should be taken away from the surface. And how exactly it will be done? With the slow block. So the first block is changing certain morphology of the surface of the egg so that no more sperm can bind, and the slow block is to remove any other bound sperm from before. Okay, so the fast or transient block is fast because it's occurred within 20 seconds right after sperm enters and interacts with the egg. So it's very, very fast. I think 20 seconds is very, very less time to initiate such kind of response. So now you know how important it is to fertilize one egg with only one sperm, because they do this very, very fast so that they can prevent polyspermy. So how exactly fast or transient block can be done? This block is not permanent because it's simply a change in membrane polarization, because we know cell polarization is always maintained by the cell membrane. That means the presence of the ions in and out of the cell membrane, that is positive ions in and out of the cell membrane. For example, sodium ion. Sodium ion is always present in the ocean, because here we are talking about the sea urchin fertilization. So while right after the fertilization is done, right after the sperm delivers its nucleus inside the egg, that causes a rapid sodium influx. So sodium start to take entry from the environment inside the egg, and that sodium influx is the primary, or what we can say, very fast response for a polyspermy prevention, because due to the influx of the sodium, a lot of secondary reactions start to occur, a series of cell signaling process start to occur inside, and that further will cause the secondary, or which is known as the slow block. Now let's look at how exactly this process works. In this case, sodium influx, sodium is transported inside. So as a result of sodium influx, the net polarity of, what you can say, of the membrane, and but we can calculate this polarity of membrane with action potential, right? Membrane potential. So this membrane potential usually is minus 70 millivolt, right? So it jumped from minus 70 millivolt to almost plus 0 to plus 10 millivolt at least. So this huge jump of 70 to 80 millivolt voltage difference is only caused due to the rapid influx of sodium ion. So once sodium ion is inside, that causes the phospholipase C to be activated. Once phospholipase C is activated, phospholipase C, what is the feature of phospholipase C? I told you that here you will see the application of cell biology in every single step, and this is the example of cell signaling. So you know phospholipase C mediated cell signaling. What was that? Phospholipase C cleaves PIP2 into diacylglycerol and inositol triphosphate, or IP3. So once they produce this diacylglycerol and IP3, IP3 causes release of calcium from the endoplasmic reticulum normally. So it increases again the release of calcium from the storage of the calcium inside the cell. So as a result, cytoplasm have more calcium now. So this more calcium will trigger the release of calcium associated or calcium mediated granules, which are present inside that egg, known as cortical granules. Okay, so in the egg they have these vesicles filled with we call them cortical granules. They produce a lot of, again, hydrolytic enzymes, which also helps in this preventing polyspermy, especially in the slow block of polyspermy. Now the question is, just like the acrosomal process we saw in case of sperm. In sperm, acrosomal process is there where they have a lot of hydrolytic enzymes, which is required for them to digest a membrane and insert the nucleus inside. While in this case, cortical granules are required for the slow block to polyspermy. So this is one side of the deal, because phospholipase C produces two components: inositol triphosphate, trisphosphate actually, and diacylglycerol. So diacylglycerol now will activate protein kinase C. So once it activates protein kinase C on the other hand will allow the activation of a proton pump that will pump out proton outside the cell, outside the egg. So as a result of which, what will happen, as protons will pumped out of the egg, it causes the pH to increase inside the egg. And while the pH is increased from the acidic environment to near neutral environment, that near neutral pH is the perfect onset for the translation of proteins, which will be required for the process of DNA replication, different enzymes definitely. And DNA replication means this is in the onset of cell division. Got it? Because this egg now is being prepared for the cleavage process of the embryonic development, right? So a lot of things occurred right after the fertilization, and it is very fast. You can see the first block is within 20 seconds, although the second block or slow block is from 20 to 60 seconds, but it is permanent because it will stay for a while. So this cortical granules, remember, while they release this cortical granules, this causes three things. The release of the cortical granules causes the swelling of the perivitelline space in sea urchin we are talking about. it causes the hardening of the vitelline layer and clipping of the sperm binding receptor. So what does that mean? It means as they swell up the perivitelline space and hard, it make the vitelline layer very hard, so no further sperm can take proper entry inside the egg. This is one thing. And second thing, they also start to clip all those binding receptors. Clipping binding receptor means kind of masking agents which will not allow any further sperm to bind with the sperm receptor here or binding receptor here. Okay, so this is slow or permanent block that is placed here. Okay, so the complex signaling process, it starts with the sodium influx, fast block. So you can see that at the end it causes the increase in pH inside the egg, and this increased pH inside the egg is the onset of protein synthesis. Okay, so protein synthesis means obviously those proteins will be required for the DNA replication, and then the DNA replication will begin. So now let's talk about the process that work after the polyspermy prevention. While the cell's pH is near neutral, which is exactly optimum for the DNA replication as well as the cell division. So cleavage simply means cell division in a specific pattern. There is a pattern of the cell division. Okay, so pattern means if you have one cell, you can divide this cell multiple ways. You can divide it like this or like this or in a different angle. So there are different ways a cell can divide itself. Now another interesting fact about the cleavage is that one cell, when it divides into two, we always draw things like one cell divides into two and they are separated, but actually cells never separate like that. Cells stick to one another. So again, how exactly the cells will stick to one another, how exactly they will stack upon one another is another pattern that is also a part of cleavage. So cleavage not only means cell division, it means the way cells are present after the cell division is also a part of the cleavage. Okay, so let's talk about the different patterns of the cleavage and the regulation of cleavage. Now, as you can see in this picture, these are all microscopic images of the egg which is represented, and you can see that this is one cell. While the cell is divided into 4 cells, this is not actually 4, this is 8 cell stage. We can also only see the 4 cell because the cells are present something like this. 4 cell, and on top of that another layer of the cells. So that's what we are talking about, the layer of the cell, which are very, very important. Okay, so here this 8 cell stage we can easily see, and you can see that the size of the cell that was present as single cell and 8 cell is not that much of a difference. They are very much similar, although they are divided into 8 cells and they stick to one another. Right? They are sticking to one another. They are not totally separated. And then again, during once the modular stage is over, we are producing. The states start to divide more and more. So cells start to surround and form a center cavity, forming the blastular stage. Here you can see that the cells are also stick to one another. It's a very compact structure. The center is prepared and it will be filled by fluid. So this, at the end, as you can see, there are also multiple cells here, but how they are arranged? They are arranged in so compact structure. It looks like as a ring, and there is a center in the middle. Okay, so this cleavage has multiple patterns. Okay, and also it has features in terms of cell division. Cleavage is not any normal type of cell division. Okay, because in cleavage we don't have normal stages of cell cycle like G1, S, G2, not like that. Actually, we don't have the growth phases at all, because if you have growth phases, it will take a long time. If you consider the stages of cell cycle, if you recall that class, we've seen earlier that G stages or growth phases are the maximum or elongated phases, right? Maximum time taken by the growth phase. But at this point we need rapid division, very fast cell division, which is not the usual way cells divide. So in this case, to divide fast, the cell have only two phases. It has the synthesis phase and the mitotic phase. So S phase means it produces the DNA, synthesized DNA, and M phase mitosis. Okay, skips all the G phases. So that's why the cells can divide that fast. And second thing, the axis of the cell division is another very important thing. These are the two most important thing about the cleavage: the cell division stages that they skip, and the second is the axis of the cell division. The axis of the cell division determines how exactly the cells are going to be arranged, they are going to be balanced. Okay, so for example, it also, this axis of cell division also depends on two factors: the way the egg yolk is distributed, and the second thing is the arrangement of cytoskeleton while the cytokinesis is working, or the cell cytoplasm division is working. So for example, not all the cells, not all the organisms have yolk filled egg. For example, amphibians have yolk, birds have yolk, but mammals do not have yolk in the egg. We don't have that. Now if you look at cleavage in frog, what you can see, it's almost like a equal distribution of the yolk content. So what we can see in the frog, the division always works like a straight division. Means, the axis is predetermined, vertical axis division, as you can see in this picture. First division vertical, second division also vertical, okay, in this angle. Third division is horizontal, and something like that. So it always continues like a proper way of dividing the cells at one direction. Okay, it's like you take a watermelon and slicing that watermelon. You always should take this analogy to explain this process. You have a watermelon, you are splicing it with the help of a knife. Okay, you can do multiple ways, but ultimately you are dividing it, keeping that same volume. You can see that volume is the same now. The number of cells being changed, is being altered. But after some time, after once it's entered into the blastular stage, then the cells start growing, even it will be more. So this was one example of the cleavage pattern in the frog. Frog cleavage pattern is known as a radial pattern of the cleavage.